Dzisiaj naszym gościem będzie Francesco Bacci, który poprowadzi webinar o nieinwazyjnej technice do ilościowego określania właściwości biofizycznych sferoid bez barwienia. Od razu proszę wszystkich na czas prezentacji wyłączyć mikrofony. Pytania można zadawać podczas całej prezentacji na czacie, a na koniec zostaną one zebrane i zadane naszemu prelegentowi. Webinar będzie prowadzony w języku angielskim i teraz właśnie się przełączymy. So yeah, welcome everybody to the webinar series Knowledge Has Layers. I'm happy to welcome here Francesco from Cell Dynamics. Everyone can ask questions about the presentation on the chat during the whole webinar, and then we will gather them and Francesco will answer that questions. So yeah, let's start. The floor is yours.
Many thanks, Anna, for the kind introduction and for inviting me to sit in this event organized by Cygnus New Technologies. I am Francesco Bacci, application specialist at Cell Dynamics. Today we have exciting news to share with the audience about our recently released technology, the Weight Physical Cytometer, that is basically a label-free, non-invasive technology for the characterization of physical properties of spheroids, organoids, 3D cell cultures in terms of size, weight, and massivity, so the compactness of this sample. So let's start with a brief introduction about Cell Dynamics for everyone who has never met us before. In a nutshell, Cell Dynamics is a biotech company born 7 years ago in Bologna, in Italy. Most of us come from academia. Our main field of expertise regards microfluidics. We develop microfluidic devices for the analysis of 3D cell culture, especially spheroids and organoids. Our technologies are hybrid since they combine microfluidics with electronic, optics, and software. And today we will focus our attention on the Weight Physical Cytometer. We have already established a nice network of distributors that covers a large part of the main states, and Cygnus is the last one. We are very happy for that. So for everyone in the audience, feel free to reach out to us directly or to our local distributor in Poland, Czech Republic, and Slovakia. So let's just move on to the reasons behind the development of our concept, this physical cytometry. So the ability of measuring the biophysics, the biophysical properties of 3D cell models. When we work with 3D cell cultures, as in this case with 3D spheroids, we observe that the spheroid formation follow multiple steps. After the initial cell seedings, cells begin to aggregate each other, and after a delay period for the synthesis of the protein involved in the cell-cell junction, they form a compact 3D spheroid with tighter connection. So I know that most of you in the audience are already familiar and used with this kind of mechanism, but I'd like to put the focus on how these objects are evolving also from the physical point of view, in terms of size, in terms of weight, and in terms of mass density, so their compactness. If we look at the picture on the left, that is taken from a paper, you can see the images of spheroid grown in U-bottom ultra-low attachment multi-well plates and acquired at different time points in bright field microscopy. And there are basically two parameters we can use to classify them: the cell number and the culture stage. We see that samples undergo a compaction phase from day 1 to day 2. It means that they are creating tighter connection, they are getting more compact. And after that, they start in size, but the system very fast reaches a plateau phase. It means that if we only look at the diameter of this sample, we are missing a part of the story. If we are only measuring the diameter, we can see that from day 5 to day 13, samples are not increasing so much in size, but they are getting darker. So something is changing. It can be the weight, can be the thickness, the viability, the mass density. So far, researchers haven't got available technology to measure and quantify the physical properties of these samples. But what we know also is that biophysics holds a biological relevance. It means that the size and the compactness of the spheroids influence also the formation of diffusion gradients of oxygen, nutrients, and also basically drug diffusion as well. So they can be a source of variability. And definitely measuring these physical parameters out of your 3D cell culture is definitely worth the time and the effort. And during the recent years, spheroids have also become increasingly important as building blocks for the field of tissue engineering and 3D bioprinting. Spheroids represent also attractive vascularization units because of their high angiogenic and vasculogenic potential. They can be printed in close proximity, and they can later on assembled into larger microtissues through the tissue fusion process. However, in order to bioprint human organs, it is necessary to develop technology for the scalable production of millions of these tissue spheroids from one side, and also we need also automated technology for the control of this fusion process. So new technologies are required to characterize and standardize the spheroid production. So far, the characterization of these spherical aggregates rely only on the image-based measurement of their size, of their morphology, the volume prediction, and researcher, in addition, can run fluorescence-based in vitro assay to estimate the viability of these samples, and they can also measure the biomechanical properties in terms of tensile strength or compression tests with AFM probe or micro tweezers. So we are trying to move in this direction. We are trying to work with enabling technologies for the characterization of these samples. As we have said in this slide, it's crystal clear that spheroids kiss as well when we use them for the 3D bioprinting, but the question is, under what conditions? They have to be viable, spheroids need to be compact, as round as possible, homogeneous, and possibly with a diameter lower than 250 microns in order to prevent the formation of the necrotic core inside the spheroid. So what is really important is to study the tissue fusion. It is a ubiquitous process that also happens during embryonic development, and that can be recapitulated in vitro. And past experiments have explored the analogy of this cellular aggregate fusion with the fusion of liquid droplets. Okay, and according to this theory, the main parameters that determine the fusion process are the viscosity and the surface tension. The problem is that cell aggregates as 3D spheroids are much more complicated than liquid drops. And this will affect the limits of the liquid drop model applicability. So if we look at the problem from another point, also the cell viability, the cytoskeleton, the cell adhesion receptors, the ECM proteins, these are factors that are essential for tissue fusion process. We have to keep into account these problems, these features, because they can change kinetics or impede the fusion of the tissue spheroids. So it's very important to measure the material properties of multicellular spheroids. And our system, our weight technology can provide a complete access to the weight, the size, and the mass density, so the compactness of these objects with a label-free, non-invasive method. And the goal is, of course, to standardize this process in its entirety and better understand how spheroids behave during this fusion process. Okay, so this is the solution we are proposing. It's called the Weight Physical Cytometer, and it's the only fully automated platform for the physical characterization of 3D sphere-like biological samples as spheroids and organoids in terms of weight, size, and mass density. Okay, the mass density is the ratio between the mass and the volume of your object, and it's an index of spheroids compactness. The technology is flow-based and relies on a couple gravimetric and dimensional analysis. This device is really small, is only 45 centimeter wide, so it can also be placed under the biological safety cabinet to work under a sterile condition. And behind this transparent safety door, we have two compartments. In the upper part, we have the weight chip compartment for the loading of our proprietary chip for the analysis of the samples. And then in the lower part, we have the liquids and buffer compartment area. So the sample and liquids holder area is designed to host 4 centrifuge tubes. And starting from the left, we have the first tube, that is the vessel A. Is a 15 milliliter centrifuge tube that contain the target spheroids that the operator wants to sort and recover after the analysis. The third tube from the left is the vessel B. It's a 15 milliliter centrifuge tube that contains all the contaminants, let me say. So the unwanted spheroid that are not recovered in the vessel A, these spheroids are recovered in the vessel B. The second tube from the left is the sample holder tube. So it contains the sample suspension under testing. So spheroids are collected in this tube. They are analyzed in our weight chip. And then the target spheroids are collected in the vessel A. The fourth tube from the left contains the weight analysis solution. That is our proprietary salt solution calibrated in terms of viscosity and mass density. And then we have the waste button. So, long story short, the device acts likewise a common flow cytometer. So, sample suspension is withdrawn from the sample holder tube by a pumping system, and individual spheroids are driven one by one inside of the weight chip that is observed by an imaging setup. Samples are driven just above the field of view observed by the 2 cameras. And in that moment, we stop the pumping system and we let the spheroid fall down only by gravity force. As such, the weight software tracks the spheroid, tracks and monitors the single spheroid during their free fall motion across the field of view only by gravity force. This is basically sedimentation theory. We are measuring the free fall speed of our spheroid, and based on the physical law, that is Stokes' law, we are able to measure the mass density of these samples that is directly correlated to its free fall speed. So the camera takes images, takes bright field images every 100 milliseconds, and the image analysis software is also able to measure the diameter and predicts the volume of our samples. So we have the measure of the mass density, we have the measure of the volume. In the end, we can extract the weight value of these samples by the ratio of mass density and volume, and we determine the weight in nanograms. After the measure, we can recover our target population of spheroids in the vessel A in terms of ranges, pre-selected ranges of diameter or mass density. All the basic principle is precisely described in this paper, so you can directly reach out to Anna and his colleagues from Cygnus New Technologies to receive the full text of the paper that entirely describes the method of our device. So at a glance, our weight system collects three outputs: the size of our samples in terms of diameter and volume, the weight, and the mass density. The device has been calibrated in terms of precision and accuracy with calibrated polystyrene bits, as we do in flow cytometry. As we said before, it is able to sort and recover target subpopulation of 3D samples. It works in this particular working region. We can analyze from 50 to 500 microns in diameter of samples, so from small clusters up to larger spheroids and organoids. And we repeat the measure for every single sample up to 10 times. It means that we can analyze the same individual spheroid falling down across the field of view of our device up to 10 times. This helps us to have mean values, standard deviation, and to get a more robust result. And based on the standard deviation, we can refine our population of samples under testing from the outliers. For example, from poorly aggregate of cells, from ellipsoid, from disaggregated spheroid, but we don't want to consider in our population. Weight represents a paradigm shift in the existing way of doing in biomedical research. Okay, so we are really familiar with this kind of workflow. Up to now, we grow 3D cell cultures, and later on we expose the samples to our drug solution, and we typically evaluate the in vitro drug efficacy in terms of variation in size, so diameter shrinkage, and viability assays. And from now on, we can add additional insights in this context. For instance, mass density represents an index to evaluate the spheroid compaction and its maturation throughout the experiment. As regards the drug testing, these physical outputs can be used also as label-free readouts of in vitro drug efficacy. So we can evaluate the efficacy of a drug in terms of weight loss of your spheroid. This a label-free quantification of the cell death, so of the drug cytotoxicity. We can evaluate the drug efficacy also in terms of diameter shrinkage, of course, but most important in terms of density variation. So a variation of the compactness of the spheroid that is directly linked to the efficacy of our drug. And we have demonstrated that there is a good match between the results coming from our technology with the outputs coming out from the well-used and well-common in vitro viability assays. So let's now take a look a little bit on the experimental workflow. In first instance, of course, there is the 3D cell culture. There are several methods on the market for 3D cell cultures. Growing spheroids and organoids in ultra-low attachment, in suspension, so in ultra-low attachment multi-well plates is the easiest way, but there are some factors to take into consideration. As the situation currently stands, we ask customers to analyze as a sample input from 20 to 100 spheroids for each test condition. So we need from 20 to 100, and thereby micro-pattern plate and flat bottom plate represent the best compromise, because with these technologies, with these consumables, we can have from tens to hundreds spheroids in each well. It means that we can directly transfer the spheroids with one single pipetting step from the plate well inside our sample holder tube. And micro-pattern plate are the most recommended consumable plates because they can deliver a highly homogeneous population of spheroids compared to flat bottom plates where we can have heterogeneity in the size and in the shape of the spheroids. So our best-in-class products are this one, we have already tested all of these 3 plates from Kugelmeyer's, stem cells, and Corning. But customer can still keep on going, keep on working with the other methods. After that, we are ready to start the analysis. So the starting point is to group and transfer from a minimum of 20 spheroids to a maximum of 100 spheroids related to the same test condition inside our 15 ml centrifuge tube, that is the sample holder tube, and resuspend them in 7 ml of weight analysis solution, our calibrated buffer. We generally analyze at least 10 spheroids for each test condition. This is the minimum number we suggest our customer to have a robust value for the statistical analysis. So after that, we are ready to insert the chip in its particular lodging. We load the buffer, and we are ready to start the analysis. Our analysis consists of 2 phases, of course. The automated image-based analysis of the physical properties of our spheroids, and the sorting procedure, the physical-based sorting procedure of target subpopulation of samples based on ranges of diameter or mass density. And now I would like just to give you a brief overview of our Libra software. Libra is our research use-only software that runs in our device. And to start a new experiment, the first screen is basically dedicated to the experiment settings. So at this stage, the user types the experiment name, sets the number of samples to be analyzed for each test condition, and then you can also set the robustness, so the number of test repetition, of measurement repetition for each spheroid, and you can also define the size range, so a rough pre-selection of the diameter of your samples under testing. After that, it is possible to proceed with the sorting options. So the operator will define custom ranges of sample diameter or mass density in order to sort and recover the target subpopulation in the vessel A. And after that, we are ready to start the experiment. This is the control panel, so the overview of our system. We have 2 cameras that are visualizing the field of view of our weight chip. We have the control panel for the procedure. We have the panel area for the real-time show of the results in terms of diameter and density. And then there is the overview section with the timer of the experiment, a brief recap on the sample information and on the procedure ongoing, and then the number of spheroids that are already collected in the vessel A or in the vessel B. So we start with the fill procedure. It's a priming procedure. We fill all the microfluidics inside the weight chip with the weight analysis solution. If any bubbles, any air bubbles are present in the weight chip, they can be removed at any time during the experiment, simply by clicking pause button in the control panel and then selecting the remove bubble function. So after the filling procedure is completed, we click on measure and then on start, and we are ready to start the analysis. So samples are withdrawn from the sample holder tube and they are driven inside the weight chip. If more than one spheroid is already introduced in the weight chip analysis area, the system will automatically separate them. Only the first one in the row will be directly considered and transported in the weight analysis channel. So just to give you more information, this rectangular shape, in this rectangular area, is delimited with these white lines. Here we have the measurement of our diameter, of the diameter of our samples, while in the low quality camera, so in this bigger analysis region, here we will measure the mass density. So the fall speed, the free fall speed of the single spheroid that is falling down. So basically, we are transporting the single spheroid in this region. There will be a centering phase. So the flow channel architecture will generate microcurrents to put the sample in the middle of the channel. You can see that. And when the sample is perfectly centered, we will start measuring the diameter. Okay, you will see a yellow circle that describes the boundary of the spheroid, and we measure it 2 times. In that case, because we have selected 2 repetitions, you can see from the overview. And after the measurement has been done, the results are immediately displayed in the graph diameter. Upon completion of this measurement, we are ready to measure with the low quality camera in this larger analysis region. We are ready to measure the free fall speed. Also in that case, we repeat the measurement 2 times. So we are analyzing the single spheroid that is falling down across the field of view. It is depicted in green with a green circle while it is falling down, so while it is under measurement. And after that, we can sort and recover it by using the right channel branching from the right. I would like just to, these operations are all automated. So you see that these operations are all automated. So all these measurements are done automatically, and results are real-time displayed in the graph diameter or density area. I would just like to give you a little highlight on the sorting. You can see that our spheroid has been collected in this additional channel branching from the bottom right. Okay, and it is collected in a vessel A. Okay. Of course, results. Of course, we have seen that results are already updated and uploaded in our software, but basically the operator can also recover them as an Excel spreadsheet file, and we suggest to show the data in a box and whiskers plot. Okay, this is probably the most visual graphic representation of our data. Potentially, every type of sphere-like cellular aggregates meets the sample requirements for the weight physical cytometer, but 3D cell models can be really heterogeneous. So we have defined compatibility criteria. We have 4 requirements. We need to have samples from 50 to 500 microns of diameter. They need to have sphere-like morphology, so they have to be quite circular. We also need to have a stable suspension of spheroids or organoids without remarkable aggregation in clusters. And it would be better to have size uniformity of the 3D samples related to each test condition. Of course, if we are testing 2 different test conditions and we have heterogeneous spheroids in size related to the same test condition, probably this effect will cover a little bit any statistical differences among the 2 test conditions if present. In the chart below, we have classified all the different sample types that we have analyzed already with our device. So we have analyzed a good number of tumor spheroids of different cell composition, also stem cell spheroids from primary cells, and we have under testing airway organoids and colorectal cancer organoids. So we have the last 10 minutes to discuss a little bit more about the application of our technology. We have identified 4 main sectors. The first one is basic research. So the weight physical cytometer, of course, provides an innovative quality control assay to standardize 3D cell models and to optimize the protocol. And I can show you some data about that. These are results already published on Frontiers in Immunology. We have leveraged on our technology to establish this label-free biophysical assay for the quality control of these colorectal cancer spheroids made up of 4 different cell lines. We have grown these spheroids in flat bottom ultra low attachment multi-well plates, and after 6 days of culture we have analyzed them under a bright field inverted microscope. And as you can see, under this preliminary investigation, it seems that HT29 spheroids show irregular and rough shape, while the other 3 cell types generates spheroids that are more round and seems also to be more compact. And this is really interesting because even if we start from the same cell seeding number, from the same cell concentration, the different cell type will generate 3D spheroids with differences in their morphology, in their size, and in their physical properties. And we have measured this and quantified this variation. As you can see from the graph below, SW620 and the LD1 spheroids show a higher weight compared to HT29 spheroids, a higher diameter, and also they are more compact. They exhibit a higher mass density. So these results are direct quantification in line with the results from bright field microscopy, and they also match with the results coming out from 2 other in vitro tests. We have indeed measured the number of cells in 1 square millimeter area with a confocal microscopy, and we have also quantified the ATP content, so the viability of these spheroids with cell titer Go 3D from Promega Life Science. And what is really interesting is that the mass density is directly proportional with the number of cells within the spheroids and with the ATP content. Also, in these 2 last graphs, the SW620 and the LD1 spheroid exhibit the higher number of cells composing the spheroid and the higher ATP content. So we can basically draw the same conclusion. This is really important also to define the most promising cell line if you are testing more than one to generate your spheroid. So this is a quality control when optimizing protocols. And let's now moving to the next application, that is cell factories. So gathering information about the spheroid's size, weight, and mass density is also important for cell factories when we want to scale up the process. So we can measure our spheroid population at precise intervals of time, over time, and we can monitor the growth kinetics in terms of physical properties. In this case study, customers have cultured spheroids composed of keratinocytes in ultra-low attachment agar-well plates from Stem Cell Technologies up to 6 days. Samples were fixed with PFA 4% with their protocol and then shipped to us in Italy. And we have analyzed spheroids at 4 different time points: 1, 2, 3, and 6 culture days. And what we have seen is in accordance with what we have said before. If we look at the third graph on the right, the diameter, we see that spheroid passing from day 1 to day 2 decrease a little bit their size. This is the preliminary compaction phase. Samples are still aggregating each other. They are forming tighter connection. And after that, from day 2 to day 6, the spheroid starts increasing its size. Okay, and this is in line also with the weight graphs. In the first phase, in the first 2 days, some cells are dying, but the other one are aggregating into spheroid. Okay, so after this preliminary compaction phase, cells start doubling and replicating, so the weight of your samples is increasing after the replication phase. What is really interesting and captivating is the mass density value. We see that there is an increase in the mass density in the first compaction phase, and after that, the mass density, so the compactness of our sample, is decreasing, basically because keratinocytes start with the secretion of soluble factors of laminin, glycans, and all the proteins in the ECM, in the extracellular matrix. And these proteins are creating a framework, a structure that creates something like a, let me say, a physical distancing between the cells composing the spheroid. This is really nice. These results will be published this year. So we can keep on track of all these parameters and how they evolve over time with this method. Another important application is drug testing. As we said before, along with the protocol standardization, the physical parameter represents also a quantitative output for the quantification of in vitro drug efficacy. These results have been submitted to a scientific manuscript. They are under review. We have grown colorectal cancer spheroids made up of Lovo cells in ultra-low attachment L-plasia plates from Corning Life Science for 7 days. After 7 days of culture, we have exposed them for 24 hours to an anti-cancer compound, a chemotherapeutic. We did image analysis with bright field microscopy and confocal. We analyzed the total protein content, and we analyzed spheroids live and fixed spheroids with our device. And these are the results we have got. After a preliminary investigation with bright field and confocal microscopy, it seems that, okay, spheroids decrease their size, their diameter, after exposure to 2 different concentrations of the drug. But spheroids after drug exposure seems also more compact. If we look at the border of the spheroid, we see that when they are treated, they seem to have more regular borders. So it seems that we have something like a collapse of the spheroid on itself after drug exposure. And the qualitative results from the imaging are confirmed also with the data coming from our device. If we look at the results on live spheroids, we see that passing from the positive control to the treated sample, we have a significant reduction in the diameter of the spheroid and a significant reduction in the weight. So we have the weight loss because it's directly linked to the cytotoxicity of the drug, and so to the apoptosis of the cells composing the spheroid. And these results are conserved and preserved when we pass from live spheroid to fixed spheroid. This is a validation of our method of spheroid fixation with PFA 4%. So if there are any statistical differences, we are able to see them also when working on fixed samples. What is really interesting is the increase in the mass density. It seems that after drug exposure for 24 hours with 2 different drug concentrations, it seems that spheroids increase their mass density. And this is also interesting because it's agrees with the preliminary investigation in bright field. It means that we have a spheroid collapse on itself. Spheroids are decreasing in size, in diameter, are decreasing in weight, in the mass. So our ratio, mass density, is the ratio between mass and volume. And we are telling you that the decrease in the volume is proportionally much higher than the decrease in the mass, in the weight of our samples. So in the end, this ratio, this mass density ratio between mass and volume is significantly increasing. It's increasing because the decrease in volume is significantly higher than the decrease in the weight. And this is basically an additional overview and insight on the mechanism of actions of the drugs. We can get to the same conclusion even if we work on a label-free, non-invasive method of analysis. And the last slide for today, before the Q&A session, is the cell therapy application of our device. The weight system can be applied also for evaluating the in vitro efficacy of cell-based therapies. For example, when we test immune cells, in this case NK cells, to treat tumors. These results are also included in the last paper released in Frontiers in Immunology from Cell Dynamics last December in collaboration with a couple of immunologists here in Italy. We grow spheroid of colorectal cancer cells in flat bottom plates, and then we expose them to NK cells in ratio 1:1 for 6 and 24 hours. Samples were analyzed also in that case fixed with PFA %, and these are the results we have got. We tested two different cell lines, SW620 and DLD1. So let's start with the first row. What we have seen is that after exposure to NK cells for 6 and 24 hours, we observe a significant reduction in the weight and in the diameter of the spheroid. It means that NK cells are attacking the spheroid and they are promoting the cell death. So we evaluate the cell cytotoxicity in terms of weight loss and diameter shrinkage. And these results are in accordance with the reduction in the fraction of living cells that is measured with a commonly used in vitro viability test. What is really interesting is the variation in terms of mass density. It seems that after the addition of NK cells and after the exposure for 24 hours, it looks like that the spheroids are increasing their mass density. They are getting more compact because of the infiltration of NK cells. But this variation is not statistically relevant. But then we repeated the test with the DLD1 spheroid. And what we observed is something really interesting. Also in that case, we have seen a significant reduction in the weight at both time points, 6 and 24 hours of exposure to NK cells. And also in that case, we have a significant reduction in the diameter. And these results agree with the outcome from the in vitro viability assay. So also in that case, we have reduction of the viability of the spheroid. What is really captivating here is that we have a statistically significant increase in the mass density of our spheroid after the addition of NK cells. So it seems here we have demonstrated that the NK cells that are infiltrating in our spheroid are significantly increasing their mass density. And we decided to investigate the reason behind this increase in mass density with confocal imaging. We have imaged spheroids with infiltrated NK cells labeled in green, and you can clearly observe that a significantly higher number of NK cells can infiltrate DLD1 spheroids throughout their entire volume, and this can be responsible of the statistically significant increase in mass density. While instead in SW620 spheroids, NK cells accumulate only at the periphery, so we have a slight increase in mass density, but it is not significant at all. So this data might explain the differences in the infiltration rate of NK cells that is quantified in terms of mass density with our device. Okay, so thank you very much for your time today, and to Cygnus New Technologies team for the organizing this course and inviting us to disclose our technology. I saved the last 10-15 minutes for the Q&A session, so I'm really excited to hear your impressions, your feedbacks, and as many questions as you have. Many thanks.
So I'll go first. Thank you so much for that whole walk through the technology. I want to inform first all of our listeners and that if you are interested in the physical cytometer weight, you can contact our representative Karolina, contact and email to her. You can find in the chat. I will also repeat it in Polish. So jeśli są Państwo zainteresowani cytometrem fizycznym weight, zapraszamy serdecznie do kontaktu z naszą reprezentantką. E-mail Państwo znajdziecie na czacie oraz zapraszam wszystkich do zadawania pytań. Yeah. Thank you very much. I think if anyone has also very specific questions, they can write directly to you, to your mail. I can't see any question now. I think that was like the big amount of information for one time. So for now, I want to thank you very much for that whole presentation and for your participation here. That was really interesting and awesome. And yeah, I will kindly welcome everyone to our next webinar series, which will take place on Tuesday. So please visit our website and see you next time on the next meetings. Thank you so much.
Thank you so much. Bye.